Hello, and welcome to the second part of part one on our muscle notes. Today, we're going to look at the general structure of muscles, both on the macro scale and on the microscopic scale. So let's take a look at the skeletal muscle. So we're taking a look right here at the brachioradialis muscle in this diagram. We can see that there's the bone, tendon attaches muscle to bone. That tendon doesn't just go around the outside of the muscle, it also goes within the muscle. So we can see structures within the muscle like this fascicle right here, this bundle of muscle cells surrounded by a perimysium, this tendon-like material around the outside of it. And then within that fascicle, we have the muscle fibers themselves, which are just muscle fibers, another word for muscle cell, and has endomysium, so more of that tendon material inside of it. The other thing we can see down here, guys, is if you ever talk about the grain of meat, like when you're slicing meat, you usually want to slice across the grain. It makes it more tender when you're cooking it. That grain of the meat is caused by these fascicles here running this direction. So the grain of this meat runs this direction, so you'd want to slice it transversely, just like we did here, if you're going to get it ready to cook for most situations. If you cut it the lengthwise, you're going to have really long, stringy bits of meat that get stuck in your teeth. Those stringy bits that get stuck in your teeth, those are the fascicles. All right, so let's take a closer look at this. Okay, so here you're looking at a single muscle fiber or a muscle cell. Okay. Each fiber is a long cylinder with multiple nuclei. We learned that in our histology unit. And they lie within the sarcolemma. The sarcolemma is a fancy word for the cell membrane of a muscle cell. Now, we give it a special name with the prefix sarco, because it has to do with muscles, because this particular type of cell membrane has some extra functions that regular cell membranes don't. It does everything the regular cell membranes do, but it also has a function in helping the muscle to contract and transmitting electrical signals, which we'll talk about in a later part of the notes. Ooh, too far. So the fibers contain all the normal organelles, like we talked about, along with something called the sarcoplasmic reticulum and T-tubules, which we'll get to later as well. But we can see here we've got the muscle fiber or muscle cell, nuclei around the outside. Within that, we see lots of mitochondria because the muscle takes a lot of ATP energy. Okay, and then within there, we have a subunit called myofibrils. These myofibrils, those are where the muscle actually does the contraction. Okay, a myofibril right here, we can see a chunk of it. And there's something in there, a substructure within even the substructure of the myofibril called the sarcomere. Sarcomere. Like I said before, I sometimes get the sarco and sacro mixed up. Anyway, so here is the sarcomere, and we'll take a look at that on the next slide. So these myofibrils, like I said, densely packed, rod-like. These are the contractile elements. So they make up most of the mass of a muscle cell. I mean, you also have a lot of mitochondria and other stuff in there, but this is the bulk of it. Okay, and if you want to get stronger, what you do is you pack more myofibrils into an individual cell. You don't make new cells. You add myofibrils to the cells you already have. And we talked about this a little bit in histology, but these myofibrils and the elements within them, the proteins within them, are so perfectly lined up that you have dark and light bands, which we call A bands and I bands, in the muscle cells. We look at them underneath the microscope. Let's take a look at that. So here's a muscle cell again, sarcolemma on the outside, mitochondria, and a myofibril. And now we're going to zoom in on the myofibril to take a look at that subsection in there, the sarcomere. Okay, so the sarcomere is the smallest individual contractile unit. We have one sarcomere from here to here, and then there'd be another one over here, and another one over here, and so on. Okay, and so that the delineating line between the two sarcomeres is this thing called the Z-disc. These blue lines here are actin filaments, which we also call thin filaments. And that is where they come together. That's where the two sarcomeres, one sarcomere over here and one here, bind up with that right here at this point. And then we got these thick red ones here called thick filaments. Those are myosin, and those are in the middle of the sarcomere. This area, I wish this thing on the bottom would stop showing up. From here to here, that is the A band, where there is that myosin filament. And that's what's going to appear darker under the microscope because we've got overlap of the thick myosin filaments with the thin actin filaments. So it's going to appear darker. And where we have no overlap, and it's just actin filaments, which are much thinner and less dense, that's where we have the lighter I bands. So that's the Z disc where they match up then to H zone. We got a little bit lighter section right in the middle here where the actin and myosin don't overlap, but it's still going to be darker because it's got the myosin in it. And then the M line is the exact middle of the sarcomere. See that again, we're just going to blow up and take a look at the actual actin and myosin filament. So we got Z discs on either end, M line right down the middle. Blue is actin, red is myosin. You can see the myosin is much thicker, made up of many myosin threads in there. And you can also see there's these little things that look like springs on the ends. Those are elastic fibers, elastin, and they're actually going to help with the muscle. You know how when you contract a muscle, it takes energy to contract it, but when you relax it, it can stretch out. You don't have to it doesn't have to push. That's because those elastin fibers are pulling it back to its resting state. Here we can see the myofilaments, the thick filaments here with the myosin molecule. Tails and the head stick up here off the side. Those will be important in a little bit. And we can see the actin or thin filament. Actin is blue, and there's a couple other parts to it called troponin and tropomyosin, which I'm going to introduce to you, but I'm not going to test you on. They are important for how it functions, but we do want to, we'll focus on the actin. Just another view of how so this would be the M line right here, right? There's myosin, actin, and the actin is going to come over to the side, and it's going to hook up to other actin over here, creating the Z disc. It's kind of off the picture, but that's how they're arranged. All right. So another new organelle we want to talk about is the sarcoplasmic reticulum. It's not really new. It's a modified version of the endoplasmic reticulum. I better get myself out of the way here. So the sarcoplasmic reticulum is an elaborate, smooth endoplasmic reticulum that mostly runs longitudinally and surrounds each myofibril. So it's running along the myofibril here on the outside of it, kind of a big old network. And what it does is it functions in regulating the calcium levels within those cells. Those calcium levels within those cells is going to be critically important for determining when those cells contract and when they relax. And we'll get to that in the next section. But the sarcoplasmic reticulum is the thing that controls that calcium level. In this next section here, guys, is where we're going to talk about contraction physiology. So what's happening chemically in there and the steps that cause a contraction. So we'll take these structures that we've just learned about and we will put them together and see how those structures allow a muscle to contract and carry out its function. Once again, structure and function rears its beautiful head in Anatomy Phys. We'll see you on the next one, guys.